Hi, my name is Shaojun Wang, a PhD student at Fudan University, and today I'm going to make an introduction about a project that we undertook to build a framework for accurate protein function prediction. So first, what is protein function? Here we use gene ontology to describe a protein function. Now, GEO contains more than 15,000 biological concepts in three domains: molecular function ontology, biological process ontology, and cellular component ontology. Protein function annotation is going to assign proper geoterms for proteins. It's obvious that proteins play important role of our life, and a protein always has multiple functions. Therefore, a protein can be associated with more than one geoterm in geo. However, knowing the function about a protein is difficult as the cost of that experiment is high and it's also time-consuming, and only less than 0.1% of proteins have valid functional annotation. Most proteins still need to be explored for their functions, so designing a high-performing method to make automatic function prediction is important, which can help researchers to improve their efficiency. But automatic function annotation is really a challenging project. First, GEO is a structural ontology. How to deal with the relationship in it is difficult, and every protein can be associated with many geoterms. As we all know, large-scale multiple label classification problem is not easy. However, there exists a huge variation in annotation. Some geoterms can annotated with many geoproteins, while some geoterms only related to a little number of proteins. So, in order to deal with the problems and make prediction more efficient, it is a good idea to use different sources about proteins to achieve high-performing prediction, such as amino acid sequence, protein-protein interaction networks, protein three-dimensional structure, and protein annotation text. Later, machine learning methods can help make predictions like logistic regression, convolutional neural network, recurrent neural network, and transformer to deal with this information and extract useful features. Based on our previous work NetGO and latest research protein language model, we propose a new framework NetGO 3 with online web server to achieve a precise prediction. NetGo 3 is an ensemble framework that integrates different sources of information. As shown in this figure, NetGo 3 has 7 component methods, where each of them utilizes one type of information. It uses learning-to-rank model to incorporate them all. Here we can see BLAST, KN, LR Swimmer, LR Interpro, and LR ESM use amino acid sequences, and NetGo use protein-protein interaction network, and LR text extracts information from biomedical literature from PubMed. We believe that learning-to-rank model can fuse these component methods effectively and make accurate prediction with the information. In NetGo 3, LRESM is a new component method that plays an important role in NetGo 3. As we mentioned before, a challenge problem in protein function prediction is figuring out how to represent protein sequences as fixed-length vectors that a computer can understand. Recently, protein language models have provided a solution that trains nonlinear neural networks for masked amino acids and extracts fundamental features of a protein with rich and semantic information. We first use ESM1B to generate protein embedding. ESM1B is a 34-layer transformer-based model trained on 250 million protein sequences. We use it to encode amino acids and get protein embedding by mean pooling. With this protein level embedding, we train logistic regression classifier for each geoterm and get a probability of which a protein has a function on the geoterm. Further, we integrate LRES with other methods and use them to run a model to fuse them together. NetGO 3 is an ensemble method which can combine the strengths of every method and achieve more precise prediction. To validate its effectiveness, we evaluate NetGO 3 on NetGO 2 dataset. NetGO 2 ranks top on CFA4. We compare LRESM NetGO 3 with baseline methods in CFA4 and NetGO 2. In this figure, we used Fmax and Smin to evaluate prediction performance. For these two metrics, LRESM outperforms baseline component methods on all three geodomains, and LRESM is better than sequence on a method that encodes proteins by LSEM. Here we also noticed that NetGo 3 achieves a better result compared to previous version NetGo 2. We believe that ESM1B is effective for constructing a more robust model, and NetGo 3 is the more superior competitive method. We list all methods' performance here and make a more detailed comparison. As shown in this table, NetKN is helpful for biological process ontology, and BLASTKN can provide help for molecular function ontology and cellular component ontology. Here we noticed an interesting phenomenon. LRESM and LRInterpro show comparative performance in all three geodomains, where LRInterpro uses protein family and domain information. LRESM show competitive performance to it. We believe that ESM1B really extracts biological information for protein function, and that's why NetGo 3 can achieve improvement compared to NetGo 2 and get the SOTA performance. Here we also explore the performance of different methods over two model species, human and mouse, to make a species-specific analysis. It's clear that all methods obtain a better prediction performance on human than mouse. LRESM achieves good performance among component methods in two species, which highly indicates its robustness. Once more, NetGo 3 achieves the best performance. To better show the strength of ESM1B embedding, we tested the ESM1B on difficult protein. We select proteins with a blast identity of less than 0.6 to any protein in training data as difficult proteins, which has little homology information. So BLAST-KN fails to perform well for these proteins. ESM based on ESM1B achieved better performance in component methods, although BLAST-KN performs worse. NetGo 3 gets the highest score in the result, so ESM1B embedding for proteins is a powerful tool for function annotation. Here we select a difficult protein as a case and show the results obtained by NetGo 3 and its competing methods. This picture depicts the directed acyclic graphs according to the relationships of 18 geoterms in biological process ontology. LRESM predicts 14 correct function labels, which is the most in component methods. With the support of LRESM, NetGo 3 achieved 15 true geoterms out of 18 predicted ones, and successfully predicted geoterms that NetGo and NetGo 2 failed to predict. We also noticed that NetGo 3 predicts geoterms in the deep layers successfully, which means NetGo 3 can predict more specific geoterms. To show the ability of prediction on geoterms in the deeper layers, we divided geoterms in the test dataset into 3 groups according to the number of annotations per geoterm. In the column 10 to 30, the geoterms only annotated a little number of proteins. These geoterms can be seen as specific geoterms and always located in the deeper layers. From this table, we can see that NetGO 3 achieved the best performance, and for these geoterms with more proteins annotated, all methods achieve better performance. So this also affirms that more training data leads to better performance. For geoterm with specific function in the deeper layers, NetGO 3 achieved the best result, which strongly suggests that ESMB is powerful for predicting protein with specific function. Moreover, we propose an online web server that can make prediction for users. To better illustrate its strengths, we compare our web server prediction results with typical web and GeoLabel to show the difference and overlap. The figures in the Venn graph represent the average number of predicted geoterms over test proteins. Typical web predicts more geoterms but achieve lower Fmax than the other two methods, indicating that false positive geoterms are common in the prediction results. NetGO 3 and other two methods differ significantly in predicting geoterms in BPO, and NetGO 3 perform better with highest Fmax. It demonstrates that NetGO 3 is more accurate and can predict more true positive terms for query proteins. Our new web server has received widespread attention. Our website has been up for 2.5 years, during which time over 4,000 people have submitted more than 1.5 million protein sequences on this site. Anyone interested in our work can enter this site to submit your protein sequences without registration and login. To better help researchers work, we provide new result page in our web server. We provide 3 ways to visualize the prediction results. Users can know the depth, prediction score, and the geostructure by clicking the button on the result page. All in all, it's obvious that protein language model helps protein function prediction, and in future we would try to find more usage to better utilize the power of protein language model. Thanks to my advisor and collaborators. If you have any questions, please reach out to me at this email.